Hi, I am Fabiola Miranda Sanchez and I am a postdoctoral researcher at the Forsyth Institute. I work with the Professor Flor Dewhurst and my expertise is in microbiology. In this presentation, I tell you a little bit about my background and my career path. I hope you like it. This is me. Currently, I am living in Boston. I have been here in the States for 4 years, but I was born and raised in Mexico City. This is my family. I have two brothers and I am always in touch with them. Last time that I visited them was in December last year. About my hobbies, I love swimming outdoors, hiking, eating tacos, playing pinball. I recently started to do bird watching. This is the last picture that I took in the JP pond, and this is a great blue heron. And recently I got a new bike and I am trying to learn how to bike well in order to commute to work. When I was 13 years old in science class, I was amazed by big characters like Anton van Leeuwenhoek. He was from Switzerland and he was the creator of one of the first microscopes. He was very curious, he was very creative, but also he kept track of the things that he was looking and seeing under the microscope. He used several types of samples, from his own fluids to drops of water. For example, in drops of water, he saw a lot of creatures that they were moving. These creatures had different shapes, different sizes, and he called them animalcules. In this video, I am going to show you some examples of the things that can live in a drop of water, in this case, seawater. See? Different shapes, different size, different colors interacting, and even you can see tiny dots that these are bacteria. This is pretty cool, and imagine being Anton van Leeuwenhoek, being the first seeing this whole universe in a drop of water. This is very cool. This is when I decided to be a scientist, in particular about the microscopic world. Before starting high school, I got my first job. My first job was in a cyber cafe. I was in charge of assignating the computers to the clients, keeping track of the printers, cleaning, mopping the floors, receiving the money from the clients. So it was a lot of fun. It was a challenge because I had to pay attention to several tasks at the same time. So I learned a lot. Another activity that I was doing before starting high school and also through the years was helping my mom on her job. Uh, Her job is to put ornaments in dresses, fancy dresses like wedding or quinceañera dresses. And as you can see in this picture, this fine work that is putting beads in a specific order, being very clean and not damaging the fabric. These are the things that I helped my mom through the years. I learned from this to be very organized, very clean, and pay a lot of attention to details. I studied college in the National Polytechnic Institute and I got a major in microbiology. I did a thesis about the soil bacteria in this place that is contaminated with heavy metals. And later I studied a PhD in biomedical sciences in the National Autonomous University of Mexico. I was studying the legume and soil rhizobia. The rhizobia are bacteria that are able to establish a relationship with legumes like soy, beans, chickpeas, lentils. So these bacteria are living in the roots of these legumes and they form nodules, and inside the nodules they are transforming the nitrogen that is in the atmosphere and transforming it in a form that the legume can assimilate. And the nitrogen is very important because it's a component of the proteins. So I was very happy, and then I moved to Boston, and I moved to Boston to do a postdoctoral stay at the MIT. After finishing my studies in Mexico, I moved to the United States and I started to work as a postdoctoral fellow at the MIT, and I was working in studying the actively growing bacterial community in an estuary. An estuary is the connection between the river and the ocean, and I was studying the huge diversity of bacteria that live there. After finishing my period at the MIT, I came to the Forsyth Institute and I am a postdoctoral fellow at Doher's Lab. I am working in studying the dental plaque bacterial community, and I am trying to find new ways to culture and isolate microorganisms from that system, the dental plaque. So, in summary, the things that I have learned are that the bacteria are everywhere. And a scientist in 1934, Bas Beckin, said that everything is everywhere, but the environment selects. What does that mean? That a lot of stuff is growing everywhere. The diversity is very huge, but factors like temperature, levels of oxygen, pressure, pH are determining who is in what place. So, if I have to summarize the things that I study, is I am studying bacterial communities and I want to find out who are their members and what is shaping their structure, what is actually saying or dictating who is going to grow in at certain moment and who is not. So that is actually pretty cool. I started to study it in the system of the legumes that I described you before. These are a drawing of a legume. You can see the root nodule. So in my PhD, I discovered that the legumes are a filter of bacteria. What does that mean? That they are going to select the best rhizobia, these bacteria that are going to help them to transformate the nitrogen. They are going to select their favorites, and that means that the host is the factor, in this case the legume is the host, is the factor that is selecting the diversity of of this type of bacteria in the soil. And in the case of the estuaries, I saw that the community that is active in 24 hours is changing, and these changes are correlated with the day-night cycle and the chlorophyll. That means that you can find bacteria that like to grow according to the level of the sun, and another bacteria that they prefer the night. Also, you can see a lot of interactions that can happen. For example, if you have a bacteria that is making photosynthesis, a lot of bacteria are going to grow as a response to that, because a lot of predators are going to eat the ones that are making the photosynthesis. So that is actually very cool, because you can see a lot of interactions happening because of the activity of others. So moving to the Forsyth, here in the Forsyth, we are very interested in the human oral cavity. By the way, this is one of the most well characterized microbial habitats of the human body. And here at the Forsyth, there is a database that is called Human Oral Microbiome Database, and it has described more than 750 species of bacteria. But all these bacteria in your mouth are not randomly distributed. Actually, there are a lot of strong associations between the oral sites and the microbial residents. As you can tell in these pictures, this is a dental plaque image and this is a tongue image, and in both of them we can see a very distinct distribution of the members. Each color represents a type of bacteria, and we can tell that they are distributed in a different way in each type of sample. For example, in this dental plaque, we can see this green color in the outside, and in the tongue we have different colors that are distributed in a different way. So this is very interesting because that means that a lot of things are happening in the different oral sites of our mouth. And well, thanks to the efforts of the database, we know that 35% of the bacterial species are on culture. So I am very interested in trying to recover more of those 35%. Thinking about how to culture that 35% remaining of the strains and the species that we can find in the oral cavity, this is a picture of a high resolution microscope, and we can see that different cells are next to each other, and there is even like this matrix of substances that is like a glue that is sticking them together. So normally, in order to culture bacteria, you put a sample of the bacteria and you put them in a liquid broth. So in a liquid broth, they are not going to be together anymore because they are going to be swimming, or maybe they are going to sink at the bottom of the tube. So these interactions that are happening naturally, they are not going to take place anymore in a liquid broth. So in my project here at the foresight, I am working with these beads. These are fish tank beads, and as you can tell, they have these pores. And here in this project, we are trying to provide with this physical structure to cells that in a liquid broth wouldn't be that close. So imagine that inside a pore you have two bacteria that are living together, and one of them is helping the other one because this yellow one is producing a good. In this case, this good is a molecule that is going to trap iron, and the iron is a nutrient that sometimes the bacteria needs. So this yellow bacteria is going to produce this substance that is going to trap the iron, and then is going to have a receptor for it. And this green one doesn't have the capability to producing that good. So if they wouldn't be together, this green bacteria would die. But inside a pore, because they are close to each other, both of them can thrive and survive. So in my work, I am working with this plaque sample, and I am going to track how is the community growing in a media without the bead and a media with it. So I am expecting that in a tube that only has liquid, I am going to recover less bacteria than a tube that has the bead, because in the bead every pore is going to put together different bacteria that could help each other to keep growing. The take-home message about my presentation is that bacterial communities are fun and they are everywhere. Through my experience, I have studied them in soils, in roots from legumes, in water systems like the estuary, and now I am very excited to study the dental plaque community. So that's it. What skills are needed to be successful in my job? I listed some of them, that they are from curiosity, observation, use your senses, make a lot of questions all the time, being organized. It's okay being a little messy as long as you keep a written track of things and don't interfere with other people's activity and safety. It's very important to receive and give feedback. The creativity is a must. It's very important that you think always in different alternatives to solve problems. You have to be a team player. That can save you a lot of time, and that can enrich your point of view and can make even more interesting your research. And you have to be very patient, because sometimes the good results and the good observations come after a long process that starts from standardizing techniques, failing several times, so be patient. Well, that's it. Thank you very much for listening my presentation. I hope that everything is okay, and I hope that all of you are safe and healthy. Thank you very much.